Once upon a time, there was a little man who was quite lucky to survive a plane crash, but then unluckily suffered from a neural injury somewhere in his peripheral nervous system, which now needs to be repaired. Of course, we can't expect to use a piece of tape to glue the nerves back together. The injured and degenerated nerves will have to do their part if they were to heal, a process known as regeneration. So today, our topic will be axonal regeneration in the peripheral nervous system. Nerves have three layers: epineurium, perineurium, and endoneurium. Nerve fibers are wrapped in endoneurium and are made of a bundle of neurons. Nerve cells have a cell body with dendrites around it. And here's the axon, which is surrounded by Schwann cells to increase the speed of signal transmission. Let's get back to this little man. While we do hope his nerves can regenerate, for that to happen, there are several criteria to be met related to axonal regeneration in the peripheral nervous system. First, when the axon is damaged, the nerve cell will be divided into the proximal segment and the distal segment. The gap between the proximal and distal sections should be close enough and should not exceed 3 mm. Also, the cut ends should be in the same line. Second, neural lemma should remain in contact because it forms a tube for axon regeneration growth. Finally, the nucleus must be intact. After meeting the criteria, how do the axons actually reconcile? Here is an important process that injured nerves routinely undergo. The proximal segment will undergo proximal segment change. There are two things happening in this change: the degradation of the adjacent myelin and Schwann cell. Also, in the cell body, nucleus migrates to peripheral from the center, and peripheral chromatolysis takes place. Nissl bodies disperse and accumulate around the nucleus. In the distal segment, Wallering degeneration happens. Wallering degeneration includes axonal degeneration, Myelin clearance, and regeneration. During axonal degeneration, the axonoma is first swelled and degraded. Inner organelles like mitochondria and endoplasmic reticulum eventually disintegrate, while the axonal cytoskeleton degrades. Ubiquitin and calpain protease actively aid axonal degeneration. The process occurs in both peripheral nervous system and central nervous system, but with a longer time in the CNS. Myelin clearance is the following step. Schwann cells respond to the injury rapidly. The MAPK is activated in Schwann cells and decreases the production of myelin lipids. Schwann cells can degrade myelin themselves or release cytokines and chemokines to recruit macrophages. The endoneurium will also release serotonin and histamine to attract macrophages. Then macrophages would identify and degrade debris marked by opsonin. The final step is regeneration. Proximal axons can then regenerate and re-innervate their targets, allowing recovery of function. Schwann cells multiply and fill the gap to form a regeneration tube for axonal sprout. The axonal sprout grows towards the distal segment in the endoneurium tube filled with Schwann cells. In this case, Schwann cells release multiple neurotrophic factors, including neural growth factors, to guide the regenerating axons. The cellular and molecular mechanisms behind axonal regeneration are complicated. Many regeneration-associated genes have been found. These include activating transcription factor 3, SOX11, and small proline repeat protein 1A. ATF3 and SPER1A promote neurite outgrowth, and SOX11 regulates neuron survival. The action behind how the regenerated nerves are guided to their original synaptic target remains elusive. A group of scientists found out that LH3 glycosyltransferase, a collagen-modifying enzyme, is required for target-selective regeneration. They subsequently observed collagen 4A5 upregulation in Schwann cells after transection. Some Schwann cells also express axon repellent SLIT1. These act as signals to regenerated growth cones and repel them from the incorrect path.